Welcome to Fun Facts Online. Today, we are diving deep into the microscopic realm to uncover a biological superpower that might just change the future of humanity. We are talking about the immortal bear. In the world of survival, there is a creature that defies the very definitions of life and death. It is not a rare bacterium or a genetically engineered superorganism. It is the tardigrade, often called the water bear. These 8-legged invertebrates, no larger than a grain of sand, have survived all 5 of Earth's major mass extinction events. They can withstand the crushing pressure of the deep ocean, the sterilizing radiation of a nuclear blast, volcanic heat, and even the freezing vacuum of outer space. For centuries, scientists have been baffled. How can an organism made of the same biological building blocks as us, proteins, DNA, lipids, survive conditions that would shred our cells in microseconds? The answer is cryptobiosis. This is a state of suspended animation where metabolism virtually stops. In this state, the water bear is indistinguishable from a lifeless mineral, yet add a single drop of water, and it returns to life, even after decades of dormancy. For a long time, this was just a biological curiosity. But that view has shattered. Groundbreaking research has recently demonstrated that the molecular machinery driving the tardigrade's resilience, specifically a unique class of intrinsically disordered proteins, can be transferred to human cells. When introduced, these proteins induce a reversible state of biostasis, effectively pausing the human cell's biological clock. This phenomenon, now dubbed the water bear effect, represents one of the most significant potential leaps in biotechnology. It promises a future where human organs can be stored on shelves, where trauma victims can be placed in suspended animation, and where long-duration space travel moves from fiction to reality. Today, we are going to tell the story of how we unlocked these secrets. Let's step back into history. The concept of suspended animation began in 1702 with Antony van Leeuwenhoek, the father of microbiology. While examining sediment from his roof gutters, he saw dried, shriveled creatures that appeared dead, but when he added water, they swelled and swam away. He wrote to the Royal Society in London saying, "The animalcules that had been dead came to life." His peers were skeptical, believing life was a continuous fire that could not be reignited once out. It wasn't until 1773 that German zoologist Johann August Ephraim Goes formally discovered the tardigrade, naming them little water bears. As microscopes improved, so did the experiments. Tardigrades were dried for decades, frozen to near absolute zero, and in 2007 sent to space by the European Space Agency. They survived the vacuum and solar radiation, and upon returning to Earth, they even reproduced. This forced us to accept a third state of existence, cryptobiosis. The organism consumes no oxygen and produces no energy, yet remains structurally intact. But how does this work biologically? To understand the water bear effect in human cells, we must understand the enemy, entropy. Life is a battle against disorder. Water acts as a scaffold for our cells. When a normal cell dries out, proteins lose their shape, membranes fuse, and salt crystals shred the cell from the inside. Tardigrades survive this through anhydrobiosis, or life without water. When drying out, a tardigrade curls into a barrel shape called a tun. Inside, a radical chemical change occurs. For years, we thought it was just a sugar called trehalose acting as antifreeze. But recently, we found the real key: tardigrade-specific, intrinsically disordered proteins, or TDPs. The most critical are the CAHS proteins. Unlike normal proteins that have rigid 3D structures like a key fitting a lock, CAHS proteins are floppy and chaotic. They look like unraveling balls of yarn. This disorder is a feature, not a bug. When water leaves the cell, the concentration of these chaotic proteins rises. They begin to interact, but instead of crystallizing, they undergo a phase transition. Think of a crowded dance floor. When hydrated, everyone moves randomly. As it gets crowded during desiccation, they link arms to form a massive web. This is fibrillization. These proteins form a hydrogel, a porous network that shields sensitive molecules and acts as an internal skeleton to prevent the cell from collapsing. Crucially, this is reversible. Add water, the gel melts, and life resumes. In 2024, a team led by Dr. Sylvia Sanchez Martinez and Dr. Thomas Boothby at the University of Wyoming took this theory and applied it to us. They engineered human kidney cells to express the tardigrade CAHSD protein. Under normal conditions, the cells acted normal. But when the researchers simulated desiccation, the water bear effect took hold. The proteins within the human cells condensed into a gel, and the cell's metabolism slowed dramatically. They entered biostasis. They were paused. When water was added back, the gel dissolved, and the human cells woke up with no damage. This was the first proof that we could borrow the water bear's shield. The mechanism here is fascinating soft matter physics. It relies on liquid-liquid phase separation. As water leaves the cell, the cytoplasm becomes salty. This triggers the CAHS proteins to stick together via beta-beta interactions, forming filaments that turn the cytoplasm into a gel. This vitrification slows down molecular motion. Since aging and decay are kinetic processes requiring movement, turning the cell into a gel essentially freezes time. Enzymes can't degrade anything, and free radicals are trapped. Unlike cryogenics, which uses cold and risks ice crystal damage, this happens at room temperature. The implications are massive, starting with the cold chain. Currently, vaccines and biotherapeutics must be refrigerated, costing billions and causing waste. The water bear effect offers dry preservation. The Boothby lab showed that factor VIII, a blood clotting protein for hemophiliacs, could be stabilized with tardigrade proteins in a dry state without refrigeration. This could democratize medicine, allowing vaccines to be shipped in envelopes to the most remote villages on Earth. Then, there is the organ shortage. A human heart lasts only 4 to 6 hours on ice. This tight window means thousands of organs are wasted. If we could perfuse a donor organ with CAHS proteins and induce this gel state, we could reduce metabolic demand to near zero. Instead of hours, we could preserve organs for days or weeks. This would allow global matching of donors to recipients and turn transplant medicine from an emergency rush into a scheduled procedure. It gets even more intense with trauma medicine. The golden hour dictates that survival drops if care isn't received quickly. Researchers are looking at emergency preservation and resuscitation. Imagine a medic on a battlefield administering a biostasis cocktail. The patient's cells enter a gel state, and their oxygen requirement plummets. They are effectively paused, buying 10 or 20 hours for transport to a surgeon. They are then rehydrated and repaired. It is speculative, but the potential to save lives is driving massive interest. Finally, the frontier of space. A trip to Mars takes months, exposing astronauts to muscle atrophy and dangerous galactic cosmic rays. Tardigrades possess a protein called DSUP, or damage suppressor, which binds to DNA and shields it from radiation. Human cells engineered with DSUP show 40 to 50% less DNA damage from X-rays. A theoretical hibernation system for Mars could combine DSUP for shielding and CAHS proteins for biostasis. Astronauts could enter a metabolic pause, requiring less food and suffering less radiation damage, making the journey to outer planets feasible. Of course, this raises ethical questions. If a person is in biostasis, are they alive? They have no brain activity or metabolism, yet the condition is reversible. It challenges our legal definition of death. Then there is the identity problem. Does the gelation process alter the delicate synaptic connections of memory? Would the person who wakes up be the same one who went to sleep? And are we playing God by integrating tardigrade DNA into our own, directing our own evolution? These are questions we will have to answer. The water bear effect teaches us that life is not a fragile flame, but a resilient structure that can be reinforced. We have stepped out of the ocean of ignorance. The next decades will be defined by scaling this from cells to organs. The water bear has survived 500 million years of apocalypses. By borrowing its secrets, we might just ensure humanity survives the next 500 million. We are finally learning how to hibernate through the hard times, waiting for the rain to return. Thanks for listening to G Fun Facts Online.